Hello, dear children. Namaste and welcome to yet another super special V.O.T.E workshop session. This is Ambika, your biology master teacher, right here on this super amazing platform of Vedantu. Well, guys, of course, I know that all of you are as excited or perhaps even more excited than we are about V.O.T.E Vedantu Online Talent Exam, which is happening on the 4 Sundays starting on the 28th of March till the 18th of April. So remember, as it's mentioned here, this is not just any other exam. It's going to be like a comeback for you, not like a comeback. It is going to be a comeback, a major turning point to realize your dreams. Yes, children. So let us get started. Today is going to be yet another V.O.T.E workshop session in which we are going to be revising the chapter, or rather the topic heredity, within hopefully not more than 45 minutes. All right, so let us move ahead, and here is an introduction about myself. Okay, so children, my name is Ambika, as I've already told you, and I'm one of the biology master teachers here. I'm a postgraduate in marine biology, and I've been working in the field of education for over 8 years now, close to 8 years now, in fact. 2013 April is when no, rather 2013 Feb is when I started my career in the field of education, and there's been no looking back. I've been exploring a lot, and here I continue interacting with lovely children like you all. So that is about it. Children, today we will be discussing these questions, okay? Rather, questions and theory related to these topics: genes and genetics, chromosomes and chromatids, alleles, what is meant by homozygous and heterozygous, inheritance, Mendel and his basic experiments, and the monohybrid cross. Of course, we'll be having quiz questions here and there, so let us completely be ready for it. Remember, it's not just for the sake of your board exams that you need to learn all this. It's for you to have a holistic learning experience so that you can make the best use of whatever V.O.T.E has to offer you. All right, children? Okay. Now, before we move on, children, there are a few things I need to ask you, a few things that I need to tell you as well. Tell me something in the chat box right now. Of course, it's a premiere session, but I would love to read those chats while it's premiering, okay? Where are you from? Which village or which city or which part of our country are you from? Yes? It's just that we want to know, because wherever you are from, whichever tiny or huge place you may be from, it really doesn't matter to us. We are just dedicated to the entire student community as a whole, right? So yes, this is about it, but it's only for us to set the mood for the session. All right? Right. Now, think about this. Was 2020 a setback to your learning? Well, I think 2020 was a setback to every one of us in the world in one or more aspects, right? And the student community has been probably one of the worst affected. And not to worry at all, I, just like many other dedicated Vedantu employees out here, the entire team of Vedantu, I have pledged for your comeback, and your comeback moment starts right now. That's exactly why we are doing this session today, right? Vedantu Online Talent Exam, as I told you, happening on 4 different Sundays: 28th of March, 4th of April, 11th of April, and the 18th of April. So, is it like you have to give all the tests? Well, it's completely your choice. You could either give one of them, or you could give more than one of them. If you want, you could take a test on all of these dates as well. And the best part is you don't need to pay anything. It's absolutely free of cost. And remember, it's an online, it's going to be an all India live free exam. Okay, all India level exam. And on these dates, it's going to be happening at 3 p.m. And the major highlights of it are mentioned here. In-depth performance analysis with an all India rank will be given to you. In-depth analysis would mean you get a much better understanding of what your strengths are and what your weaknesses are. And that, I think, is super duper important for any student at this point of time. Coming back to school, to offline classes after the pandemic, it's quite natural to feel a little lost. We are here to guide you, to help you understand where you are right now, right? Okay, and then of course, the guaranteed scholarship from a pool of over 500 crore. Imagine. Yes, such a huge scholarship pool that we have. And it's not just on these dates, there are going to be daily challenges as well to win exciting prizes worth 1 crore rupees. And this is probably the most exciting part. You get to be Vedantu CEO for a day and win a trip to NASA. Can you ask for anything better in your student life? Yes, of course. Amazing, right? Now, in-depth performance analysis, I've told you this. So these are the details about how it's going to be an in-depth analysis. As I told you, you will be getting an all India rank. Apart from this, subject-wise analysis will also be there. State-wise rank will be another feature, and question level analysis. So that means it's going to be a complete analysis so that you know where you need to focus on better to get back on track in the year 2021. Isn't that super duper cool? Yes, of course it is. So this is what I was telling you about: win exciting prizes, an all expenses paid educational trip to NASA in the United States of America. And you get to be the CEO of Vedantu also for a day. And apart from all this, win exciting prizes and gadgets like drones, telescopes, microscopes, laptops, so many more creative and super amazing, super exciting things by taking up daily challenges. All right. So this is about it, children. And let us get started. I will tell you more about V.O.T.E as we go deeper into the session. But right now, time for us to focus, because if we are organizing an exam for you, we also want to make sure that you guys are completely prepared for it. And that's why we have all these revise these topics for V.O.T.E, like V.O.T.E workshop sessions on YouTube as well, right? Okay, so today, as I told you, we are going to be discussing heredity. Genetics, it's the branch of biology concerned with the study of heredity, genes, genetic variation, and heredity in organisms, right? So it's a very, very exciting field of science. These are some of the major things that we need to know when we start our understanding in the field of genetics. First of all, we need to know what chromosomes are and what genes are and what DNA is. These are like the most commonly used terms here. Now, children, just remember, think about human beings as a huge multicellular organism. You know how many cells make up our bodies? It's approximately 38 trillion cells which make up one human body. Now, considering we are made up of trillions of cells, let us visualize zooming into one of these cells. Now open your eyes and look at the screen. Inside one of those cells, one of those typical cells, you would see organelles, which include a lot of organelles which are arranged in the cytoplasm, and the control center, which we call the nucleus. And within the nucleus, let's go a little deeper. Inside the nucleus, there is something super important. You know what that is? We call it the chromosome. Biochemically, the chromosomes are made up of what we call DNA, deoxyribonucleic acid. Segments of DNA are what we call genes. Right? It's as simple as that. Now, remember, at the time of cell division, when you look into a cell, this is how you would see it to be. 23 pairs of chromosomes is how you see it to be. But otherwise, in a non-dividing cell, normally, you wouldn't see it so organized and so systematically arranged. You would rather see it in the form of clusters of thread-like structures like this, which we call chromatin. I'm just telling you this so that just in case you come across these terms in the V.O.T.E exam or any other competitive exam, you should be in a position to answer. All right. So chromatin and chromosomes are pretty much the same, but the major difference is the fact that chromatin is this entire clumped up, the cluster of thread-like structures, which is made up of DNA biochemically. But the very same chromatin at the time of cell division gets organized into really, really compact structures, which we call chromosomes. So chromatin condenses to form chromosomes. So obviously, both the composition of both are one and the same. That's going to be DNA. And the segments of DNA are called genes. There are many different genes, right? Gene for eye color, gene for hair color, gene for hair texture, gene for skin color. So many genes thousands and thousands of genes which are found in the human body, right? So they are segments of DNA, and we can say that chromosomes are the vehicles of heredity because they carry around the genes, right? Okay, now chromosomes and chromatid, these are two other terms that possibly you might get confused about. So as I told you, at the time of cell division, this is how you would see a chromosome to be, right? This X-shaped structure. This X-shaped structure, as you can see, is made up of two identical parts like this. This is one part and this is another part, right? You know what they are? We call each of them chromatids, sister chromatids, right? They are called sister chromatids because if you look at it at a deeper sense, they are exact carbon copies of each other. But these sister chromatids would be attached together at the center by a structure called the centromere. Centromere. So this is again something that you would find only at the time of cell division, the arrangement of the genetic material in the form of sister chromatids attached by the centromere. So chromosome is comprised of sister chromatids attached by the centromere at the time of cell division, or rather when a cell is preparing for cell division. Okay, yes. So all of these additional structures are also mentioned here. The centromeric region is here. The mitotic spindle, microtubules, you know what they are? They are specialized structures which help attach to this centromere, and another end would be attached to the poles, specialized structures at the poles of the cells, so that at the time of cell division, they can just easily pull apart sister chromatids to divide a cell into two similar daughter cells. They are super duper important. The spindle formation and all of that. All right. So centrioles and centrosomes play a major role in this phenomenon, which I just told you, this pulling apart and all of those, right? I just told you that because the term is mentioned here. No stress needed at all, just in case you're finding it difficult to understand. Not a problem at all. Okay, right. Now, next is the term allele. Now, alleles, we can describe them to be a pair of alternative forms of a gene. Alternative forms of a gene. What would that mean? For example, you have a coin. Once you're just flipping the coin and you can either get heads or tails. Nonetheless, both are different sides of the coin, right? Likewise, alleles are versions of genes. We can call them versions of genes, right? So where do you get them from? Of course, you get them from your parents, right? So to be a little, I know this diagram seems a little complex for us, may seem a little complex for some of you. Now remember, children, alleles are found on homologous chromosomes. Homologous chromosomes, which are similar looking chromosomes. Say, for example, inside a human cell, there are 23 pairs of chromosomes. Now, when I say 23 pairs, I mean it's made up of pair 1, pair 2, pair 3, pair 4, pair 5, and so on till pair 23. Say, for example, I am zooming into pair 1, chromosome pair number 1, inside a typical diploid human cell, maybe like a skin cell or something like that. I would see that one pair would obviously be comprised of two members. Where am I getting these members from? One member, I get it from my mom. The other member, I get it from my dad. So in every human being, this arrangement remains the same. Pair 1 to pair 23. So where is the difference? What makes you you and what makes me me? It is the alleles which are present on them, because we inherit the versions of genes from our respective parents. For example, I may inherit the red hair color from my mom and maybe brown hair color from my dad, right? Maybe I might inherit the tallness gene, a tallness allele from my mom, and again a tallness allele from my dad. I'm going by the example which was mentioned here, right? And these are just a few examples, but out of them, of course, only one might be expressed physically. At the genetic level, I would definitely have a copy of whatever I have inherited from my mom and what I've inherited from my dad. And this rule stands the same for every sexually reproducing organism. The difference comes in the sense that the number of chromosomes in every species would be different. That number is 23 pairs of chromosomes in human beings, every human cell. 23 pairs of chromosomes in each diploid human cell, right? So this is one major thing that we need to understand. Why do we call them pairs? Basically because every pair is formed by a copy of each of our parents' chromosomes. As simple as that. Which means for every single trait character that we exhibit, we've got hidden versions of those from our mom's side and from our dad's side. Isn't that interesting? Yes. So alleles, we can say, are alternative forms of a gene, two or more alternative forms in general, right? So this is about what alleles are. They are found on homologous chromosomes. Now, homozygous and heterozygous. Talking about alleles, as I told you, sometimes it can turn out that I inherit the brown eye color from my mom and brown eye color from my dad. From both of them, I probably inherit exactly the same kind of allele, which means my genotypic combination would become this, capital B, capital B. So you can describe that to be a homozygous condition because both the alleles are similar. And likewise, if I inherit a blue eye color allele from my mom and a blue eye color allele from my dad, again, it's going to be small b, small b. Both are exactly the same. That's also an example of a homozygous condition. What if I inherit a brown eye allele from my mom and a blue eye allele from my dad? Or the opposite, blue eye allele from my mom and brown eye allele from my dad. This would mean my genotype would be different, one capital B and one small b. So both of these conditions, capital B, small b, or a small b, capital B, pretty much they mean the same. That would be a heterozygous condition. Heteros would mean different and homo would mean similar. Heteros would be different, right? So these are some very, very important basics that we need to know. So now I think it's easier for us to understand why father and son may look exactly alike, mother and daughter may have exactly the same birthmark, and so on. Heredity and inheritance. We can say heredity is the phenomenon of passing on of traits from parents to offspring. So this could be something really good. It could be some diseases as well. Cancer, diabetes, and all of these do run in certain families. That also comes as part of heredity. Now, inheritance is the passing of genetic information from parent to child. Inheritance is more of a commonly used term in English, right? Okay, so now let us quickly get down to a quiz. Ready? Okay. Which of the following diseases cannot be inherited? Cancer, diabetes, malaria, thalassemia. Which of these cannot be inherited? Which of them is not genetically controlled? I think a couple of examples I've already told you. Your time ends in 3, 2, 1, and go. Right. The answer is malaria because malaria is a pathogenic disease, right? A communicable disease. Now, cancer and diabetes, I guess everyone knows. Thalassemia is also a genetically inherited disorder whereby there would be a problem, there would be an abnormality in the blood cells of the affected individual. Okay, right. Sickle cell anemia, thalassemia are all blood cell related disorders. Right, next one. The smallest segment of the chromosome where genetic information is stored for a particular trait. Well, let's not call it peculiar, for a particular trait. That makes a little more sense, I think. Chromosome, DNA, gene, or allele. What would you say the answer could be? Smallest segment of the chromosome where genetic information is stored for a particular trait. Your time is up. The answer is gene, segment of the chromosome. Allele is not really a segment of the chromosome. Allele would be versions of genes. But segment of chromosome, when that's the question that's being asked, of course, it has to be the gene, right? Okay. Now, Continuing further, how do we know so much about genetics today? It's because of this gentleman, Gregor Johann Mendel, who during the time when microscopes were the good quality compound microscopes were not even so popular in the world, or not even much about DNA and genes was known, chromosomes, nothing was known to mankind. This gentleman, based on his logical observations, came up with a lot of theories. How exactly? He studied some plants which he passionately grew in his garden. Now, this gentleman was an Austrian monk, Gregor Johann Mendel, and he had a pea garden of his own because he was a very, very passionate gardener. He was a very good mathematician as well. And combining these two, the logic of math and his love for plants or biology in general, he came up with the theories of genetics. So we have to be really, really thankful to Mendel for this, right? So the plants that he grew were the garden pea plants called Pisum sativum botanically. Now these were the characters that Mendel studied. So Mendel studied 7 different characters, 7 contrasting pairs of traits in the pea plant, which included all of these, okay, whatever is summarized here, because he noticed that some pea plants could be tall, some of them could be short. Some of them had axial seed pods, some of them had terminal seed pods, the position, I mean, right? Then some of them had green seed pods, some had yellow seed pods, some had a full pod, some of them had a constricted pod appearance, some produced white flowers, some of them produced violet flowers, yellow seeds, green seeds, round seeds, and wrinkled seeds. These were all characters that Mendel observed in the pea plant, and he was very, very curious to know what it was within the plant that determined which one of these was expressed for each of these characters, right? So that's exactly how he went on to his studies. Now, children, taking a very, very quick break. I told you that I'll tell you a little more about V.O.T.E as we go through the session a little deeper, right? The pattern for you guys, for this grade, for 10th graders, this is going to be the pattern. You will have a math section, mental aptitude test, and you will also have a scholastic aptitude test. Okay, so a combination of both of these. Of course, under scholastic aptitude is where our subjects would come into picture, and science does play a major role over there. The total number of questions would be 70. Right? So this is going to be the marking scheme. 4 marks for every right answer, -1 for every wrong answer. So remember this. Can be a little tricky that way, which means you cannot just attempt anything and everything that you come across. 75 minutes would be the exam duration. It's important for you to have an idea of this, children, because for every grade and for every every level, it's going to be the kind of questions and the pattern of exams is going to be different, right? Because for lower classes, for some of the lower classes, there is only going to be a mental aptitude test. For you guys, it's going to be SAT and MAT both. So are you guys excited to make a comeback? We are all definitely excited. We are all waiting to see what sort of impact it's going to make on your future. 2021 is going to be amazing just because of V.O.T.E. Trust me on this. Okay, so here is the next question. Scientific name of pea plant used by Mendel in his experiments. Yes, look at all the options and tell me the answer, A, B, C, or D. What could it be? Your time ends in 3, 2, 1, and go. The answer has to be B, Pisum sativum. You write it in italics, and of course, in all the others, it is Pisum sativus, sativis, P sativa, and all of that, right? So they don't make sense anyway. It's just a matter of one letter that's different here, right? Sativus and sativum. So good lesson for you all, so that you guys don't go wrong with it without reading the option completely, right? Okay. Now comes the next part of it, the monohybrid cross, which Mendel started his experiments with. So what Mendel did was he went on to perform heredity genetic experiments in plants, considering one character at a time. He wanted to study the inheritance of one particular character at a time. Such crosses, wherein one particular character is considered at a time, is called a monohybrid cross. It's a genetic mix between two individuals who typically have homozygous genotypes. That is how Mendel did it. But as I told you, the term monohybrid refers to considering one particular character at a time. So this is how Mendel went on to do his experiments. What he did was he chose, as the parent plants, he chose one parent that had been having a history of only producing tall plants. The other parent which Mendel chose had had a family history of producing only dwarf plants. So as you can see here, that's the parental generation, tall and dwarf. Of course, the allelic combination here is something we know about today. Back then, Mendel knew nothing about it because Mendel just observed it. Merely, he was just observing all of these plants externally. He didn't know what was going on at the genetic level. All right, so keep that in mind. And then coming to the gamete formation, remember every sexually reproducing organism, when it produces gametes, they are going to be haploid, having half the number of chromosomes as a typical diploid cell. Likewise, these parents produced gametes containing only this allele, and these parents produced gametes containing just the small t allele. When they came together in fertilization, they produced capital T and small t, a heterozygous combination, which when Mendel looked at it, it was just like the tall parent. Because phenotypically is all that Mendel could observe, right? As I told you, only the physical appearance was something that Mendel could observe. So these plants looked exactly like the tall parents. Tall. So Mendel, curious Mendel, went on to allow them to self-fertilize because he wanted to understand what role this parent would have played in such a cross. So Mendel went on to allow them to self-pollinate, and he noticed that in the next generation, which we call the F2 generation, there were tall and dwarf plants in the ratio of 3:1. 3 tall is to 1 dwarf. Today we know this is going to be the genotypic combination. 1 capital T, 2 capital T small t, and 1 small t. Right? So 3 of them are tall, 1 of them is dwarf. Yes, so this is what Mendel observed. So this would be, all right, so I think this is a typo. This will have to be the P generation. This would be the F1 generation, having all tall offspring. And this would be the next cross of the F1 generation. And this would be the F2 generation. Sorry for that typo, children, right? So the tall and short plants were crossed. This is what Mendel observed. So the ratios are very, very important for us to know. Mendel saw that in the F2 generation, the phenotypic ratio was 3:1. And today we know that the genotypic ratio is going to be 1:2:1. Right? Okay, so if you actually think of it, something very, very interesting, the genotypic ratio, I'll just write it here. Genotypic ratio is 1 capital T is to 2 capital T small t is to 1 small t. Now, if you just add these up, those two combinations which have at least 1 capital T alleles, it's this and this. When you add them up, what do you get? Yes, 3, and phenotypically they are tall. 3 tall and 1 dwarf. This is going to be our phenotypic ratio. Right? Okay, children. This is Mendel's set of monohybrid experiments. Now let us quickly try and answer this. What will be the genotypic ratio of the cross capital R small r and capital R small r? So it's written there what R stands for, round and wrinkled. What would the ratio be? Think about it. Work it out if you want, and then tell me the answer. I'll give you a hint. It's going to be exactly the same as the previous one. Okay, your time ends in 3, 2, 1 and go. How is it 1:2:1? We are talking about RR and RR, right? So the two kinds of gametes that they produce would be this, and even in this case, it's going to be exactly the same, right? So at the time of at the time of fertilization, we need the fusion of one gamete from this parent with one gamete of the other parent. So this combination, there would be this combination, we would have this combination as well as this, which means it's going to be R, 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 and small r. So genotypically, it's going to be 1:2:1. That becomes our answer, right? Okay, next one. The phenotypic ratio, tall is to short, of the F2 generation of the monohybrid cross. What would that be? Phenotypic ratio in the F2 generation. This has to be much simpler. Yes, 1:2:1 we've seen was the genotypic ratio. Would that be the same as the phenotypic ratio, or is it going to be different? Your time ends in 3, 2, 1, and go. The answer has to be 3:1 because both of these are, and then we've got this combination as well, right? And this. So phenotypically, externally, all of these would be round seeded. And this one would be wrinkled seeded. So that's going to be 3:1. 3 round:1 wrinkles, right? Okay, so if you actually paid attention to it, if you actually paid attention to it, it mentions tall is to short. So it's going to be exactly the same, capital T, this, and this. All right? Yes, so that brings us to the next one. A cross between a tall plant and a short pea plant resulted in progeny that were all tall plants because dash. Read through your options. Because tallness is the dominant trait, shortness is the dominant trait, tallness is the recessive trait, or height of the pea plant is not governed by genes T or T. What do you think could be the answer? Why did they result in progeny that were all tall plants? What's the major reason? Your time is up. The answer is A, because tallness is the dominant trait. Remember, the dominant trait, no matter what the genotypic combination is, be it this or the heterozygous condition, wherever there is at least one dominant allele, it would express in the progeny. Okay, but for the recessive one to express itself, it needs to be present in 2 copies, right? Okay, now look at this. Mendel observed 7 pairs of contrasting characters in the pea plant. Which one of these is not a character studied by him? Quickly tell me the answer. Let's number them as A, B, C, and D. Yes. Look at the first one. It shows green stems and woody stems, round seeds and green seeds is B. C would be axial flower and terminal flower, and D would be the tall plant and the dwarf plant. Which of these was not a character studied by Mendel? Okay, and your time ends in 3, 2, 1, and go. The answer is option A. Green stems and woody stems, that's got nothing to do with heredity. Plants which are tender and young would be green stemmed. Plants which are, some plants which are older and formed like a tree, they would have a woody stem, like a bark, right? All the others are, of course, those that Mendel considered. Now, next one. In human males, all the chromosomes are paired perfectly except one. What are they? Yes, pick them from the box given here to you. Large chromosome, small chromosome, Y chromosome, X chromosome. Would it be 1 and 2, 3 only, 3 and 4, or 2 and 4? What do you think? In human males, the unpaired chromosomes would be, yes, what would they be? Your time is up, and the answer is 3 and 4. Y chromosome and X chromosome, the 23rd pair is comprised of an X chromosome and a Y chromosome in the human male. As far as the human females are concerned, it's going to be 2 X chromosomes. Okay, right. Now, children, to know more about V.O.T.E, there is more that I will be giving you information about V.O.T.E. So Stay with me till the end of the class, or just in case you just can't wait for it, visit the link which is given in the description box below, vdnt.in/vdnt910e. Okay, so that's the link which is given down there. You can also download Vedantu's learning app on Google Play, and here is the Telegram link as well, so that you can click on it and directly check out all the details. Right, children, now another very, very important point under genetics is sex determination. What decides whether a baby is going to be male or female? Yes, so you must have heard about this a lot. It's a very, very interesting phenomenon, and it is only the sex chromosomes, which are the X and the Y chromosomes in human beings, which are involved in this, right? So, as far as human beings are concerned, males always have one pair of sex chromosomes, and so do females. But in the case of males, it's going to be one X and one Y chromosome. In the females, it's going to be two X chromosomes, right? So this is how things work. Among the sperms that a male produces would be two types. Say, for example, 1 million sperms are produced at a time. Half of them, pretty much, approximately 50% of them, would carry approximately 50% would carry the X chromosome. The remaining 50% would carry the Y chromosome. What about females? Of course, they produce just one ovum at a time, because every month, typically just one ovum is produced in a sexually mature female, and that would always carry only an X chromosome, never a Y chromosome. Now comes the question, which of these decides it? Of course, the egg cell is always destined to become a female, because the egg cell doesn't have the Y chromosome at all. But at the time of fertilization, millions of sperms are released into the female reproductive tract. And in that race among these millions, whichever sperm finally wins or manages to fertilize the egg, that is the decision maker. So if the winner happens to be an X carrying sperm, it results in a female. And if the winner happens to be a Y carrying sperm, that results in a male. As simple as that. XX would be the female, XY would be the male combination, right? So yes, children, now tell me this. Sex of the baby can be determined by dash. Mother only, both mother and father, father only, or none of these. I've just told you the answer, so I don't want to give you more time. Your time ends in 3, 2, 1, and go. Of course, it is the father only, because depending upon whether it's the X or the Y chromosome, this decision is made. Now, how do we summarize? What are the things we've discussed today, children? We've discussed the basics of heredity. We've discussed the basics of Mendel's experiments, right? Basics of Mendel's experiments. We've talked about the monohybrid cross. And we've also spoken about sex determination in humans, right? These are the points which we have discussed today. All right. So children, are you now all set to make a comeback? Yes. So register for V.O.T.E right now. This has to be your top priority point on your to-do list right now. Okay. So remember, you can download the Vedantu live learning app or visit vedantu.com/vote to check out more details. Remember, this is not just any other exam. It's going to be a comeback. And we've been explaining how it's going to work for you. What you do is register yourself and create your voter ID card. What exactly is this going to be like? When you register for V.O.T.E, it's very, very important to give correct details. Okay, so your name, your grade, which board you belong to, and your target exam. Why is this important? Because for every grade and for every board, the exam is going to be different. All right. So depending upon, since it's so much dependent on this, it's very, very important from your side to get the right voter ID card created so that you get access to the correct exam. Right, children? So there is your voter ID card. And after this, you can pick your vote days. Remember, it's happening on 4 Sundays, 28th March, 4th April, 11th April, and 18th of April. You can either pick one or all of these dates, or maybe 2, 3, however you wish to. Right? That's completely your choice, because it's going to be free of cost anyway. So pick your vote days and you will be done. That would be your final step. Now, Apart from this, it's not just about those 4 Sundays, right? Daily and weekly challenges will also be happening. So stay tuned. Check out all the details on the app, on the website, vedantu.com/vote, and figure out everything for yourself. These are going to be the exciting prizes which will be available for the daily and weekly challenges. Right now comes yet another interesting thing. What are squads? Now, squad, I think the word itself has to give you the hint, right? It's something like a gang. You can create your own squad and have a lot of fun together. But how do you do this? What is to be done? Definitely you have to figure out yourself. Check out all the details on the website and you will get an idea about what this is going to be. Look at all this, so many exciting things, right? Create your own squad. You can create a squad logo. You can create your own a squad slogan, something like "Samajh aayega to maza aayega to samajh aayega." Probably if we all at Vedantu could create a huge squad of all the employees together, like thousands and thousands of us together, probably this would have been our slogan, right? Okay. So, yes, children, that is about it from my side. Now, I think you are all set to get started with your preparation for vote, because as I told you, we will continue doing this hard work for you completely with full passion and dedication. And hopefully, this workshop, revision workshop, is also going to help you perform better. So, children, that's about it from my side for the time being. Please click on the like button and stay subscribed to the channel for more regarding vote and more regarding your academics. And I'll see you again. And until we meet again, this is Ambika signing off. Bye bye.